MS Ramaiah University of Applied Sciences.
Hi students, we are going to learn about staining and mounting. Aim of the experiment. The aim of the experiment is to perform staining and mounting of thin sections of crude drug. Apparatus and reagents required: Bunsen burner, watch glass, holder, glass lights, cover slips, camel hairbrush, pith needle, filter paper or blotting paper, thin sections, a dropper, chloral hydrate solution, fluoroglucinol, concentrated hydrochloric acid. Introduction. Staining is a process in which chemical dyes are used to impart color to various tissues in a section of a drug sample, which enables to distinguish the arrangement of various tissues in the sample. A stain is a chemical dye which combines chemically or physically with the cell content to impart color to the lignified tissues. Example: saffronin or fluoroglucinol and concentrated hydrochloric acid reacts with lignin to give pink color. Similarly, iodine reacts with starch to give blue color. Procedure. Prepare thin sections of crude drug. Transfer the sections to a clean watch glass. Add sufficient quantity of chloral hydrate solution and warm the section for 1 to 2 minutes on a Bunsen flame. Chloral hydrate is a clearing solution which removes the pigments like chlorophyll, fixed oils and fats and make the sections clear to view the tissue arrangements. After warming with chloral hydrate, add fluoroglucinol and concentrated hydrochloric acid in the ratio 1:1, which will stain lignin, which is a cell wall constituent. The next step is mounting. Take a clean glass slide. On the slide, transfer the section with the help of hairbrush. Add 1 or 2 drops of glycerin with a glass rod or a dropper. Take a clean cover slip and with the help of pith needle, place the cover slip gently on the section. If any air bubbles are seen, lift the cover slip and add a drop of glycerin and replace the cover slip till the air bubble is removed. Wipe off the excess glycerin present outside the cover slip with the help of a blotting paper. The slide is ready for observation under a microscope. Report. Staining and mounting of the sections of the given sample was performed.
Hi students, today we are going to learn about the experiment staining and mounting. So the aim of the experiment is to stain and mount the prepared thin sections of crude drugs. Apparatus and reagents required for this experiment are watch glass, holder, glass slide, cover slip, pithing needle and a camel hairbrush. Reagents required are chloral hydrate solution, fluoroglucinol, glycerin, concentrated hydrochloric acid and Bunsen burner. For performing this experiment, staining and mounting, we require thin sections of crude drug. So this I have prepared from this leaf, particularly from the midrib region. I have already prepared thin sections and I have kept ready for this experiment. Now thin sections are ready. Now we have to start with the staining process. To perform the staining process, transfer thin sections of the leaf into another watch glass. After transferring the thin section to the watch glass, add few drops of chloral hydrate solution. Chloral hydrate is used as a clearing agent. It clears most of the common cell contents like chlorophyll, starch, fats and fixed oil and makes the section clear for visualizing other tissues. Now switch on the burner. Light the burner with the help of a lighter or a matchbox. Now hold the watch glass containing sections and chloral hydrate with the help of a holder and warm the sections with chloral hydrate for 1 to 2 minutes. Remove the watch glass from the burner and allow it to cool. Switch off the burner. To the cleared section, now we have to add equal proportion of fluoroglucinol and concentrated hydrochloric acid. The ratio 1:1. After staining the sections with fluoroglucinol and concentrated HCl in the ratio 1:1, we can see the sections showing pink color due to the presence of cell wall constituent lignin. So lignin will react with this staining reagent and show a pink color. Now staining process is over. The next step is mounting this stained section on a glass slide. Now, with the help of camel hair brush, a thin section is taken from the watch glass, kept on the center of the glass slide, one or two drops of glycerin is added, which acts as a mount. Glycerin will keep the section fresh and avoid drying. So now I have added 1 or 2 drops of glycerin. So glycerin will prevent the drying of the section and keep the section fresh for 1 or 2 days. Now, with the help of a cover slip, I am going to prepare the mount. So hold the cover slip on one end of the mount with the help of a pithing needle. So hold the cover slip at 45-degree angle and with the help of a pithing needle, slowly release the cover slip so that the entire cover slip should be filled with the mount glycerin. If there is any gap in the mount, the gaps are filled with glycerin by adding 1 or 2 drops through the sides of the cover slip. The excess of glycerin can be removed with the help of a filter paper. So you can wipe the sides. Now the section is ready for seeing under the microscope. The mount should be free from air bubbles and completely the cover slip should be filled with glycerin. This completes the experiment, staining and mounting.
At MSR UAS, we spark your imagination unlike any other.